Hello, I'm Matthew Pocock. I'm here today to talk about ShortBOL. I currently work in Turing 8 Hamster Limited as a consultant, but I used to work in computing science with Professor Anil Waipat, and he, together with James McCullin, one of his PhD students, helped me put together this talk and contributed a lot of the ShortBOL work which is in here. So really I'm here today to talk about how ShortBOL supports the synthetic biology design process. So we should probably start off by agreeing what synthetic biology is. I know some people don't really believe it exists. They think that there's genetic engineering and there is metabolic engineering and other related practices. I don't entirely agree. I see synthetic biology as really a discipline and practice which can be applied to these things, where you rigorously apply engineering principles to producing your biological systems. So it's really important in a synthetic biology approach to major on the practice, and this includes a focus on design, on planning. You make things or fabricate them, you test them to see if they behaved as you wanted, and in doing that you make various measurements. You then refine your design and you go around the cycle again of building a better widget, in this case a better biological system. And certainly in contrast to earlier genetic engineering approaches, synthetic biology is very heavily knowledge driven. So really leveraging everything we know from systems biology, from our ability to numerically model biological systems, and from the genomics revolution, trying to design systems based upon how we know and believe they may behave, rather than an ad hoc throwing things in and seeing what happens. And the other thing I think is that through the synthetic biology approach, we're trying to raise the level of abstraction of the things we're designing away from individual genes or gene products and up to the level of whole system changes, engineering how the entire cell behaves or how multiple groups of cells communicate with each other, and potentially all the way up to eukaryotic organism design. I know a lot of this is way in the future at the moment, but this is really what synthetic biology is shooting for, as opposed to just making GFP. And in raising the bar in this way, we're really looking about how different people and different groups communicate. So the more complicated your design is, the more specific you need to be about describing it. And there's various aspects of it that can no longer be implicit once you're working with partners or once you're describing it to someone else. So there are the physical parts, obviously, and we most readily think of engineered DNA sequences, but there are synthetic biology designs that act at the level of RNA or cell lines, and then there's all the supporting stuff to make the system behave as you wanted it to. So things like the media, the food stocks, even the containers and their shapes sometimes have an impact. So that's all physical stuff. And then as well, and perhaps the more interesting thing, is the intended behavior. So in a simple metabolic intervention, you might be interested in production rates. In a biological computing environment, perhaps you're looking at oscillations of products or latching behaviors. And for diagnostics, obviously the old favorite of it changes color in response to X. But those are all dynamic things. They involve various physical components interacting over time to achieve a desired behavior. The other thing, so the two big interfaces where you need clear communication is when you're doing collaborations across organization boundaries or across groups, and when you're putting in supporting materials for publication. So if you just work by yourself on a bench, obviously you can get away with holding as much as you need to in your head and in your lab notebook. But once there are 3 or 4 different groups working, perhaps one of them is focused on fabrication, one is measurement, One is growing things to sufficient size to test, I don't know. However it works for you to break down. At the interface between each of these, you need to clearly communicate what it is you expect the other collaborators to be doing, and they need to know from you what they expect to be getting. And really you have to document the what it is as well as the what it will do. So if it changes colour in response to X, you need to not just specify the colour, the X, but you need to specify things like what is the time lag between X being introduced and the colour changing, for example. And the more distant your collaborators are, the more explicit you need to be about communicating this. If you're in a situation where there are different commercial partners, perhaps where some of your work is being outsourced to a commercial lab, you need to be particularly clear about this because they're doing a job of work and you really need to know what that job of work is that they are achieving and if they've done it or not. So the flip of this is within academic publication. Many papers have supporting materials. Most papers in this sort of area would have methods and results and materials. You often cannot reproduce the work they did from this. It's insufficient. It doesn't go into enough detail. So at the very least you need the DNA sequences, but you probably need to know media, you probably need to know, in some cases, time of day things were done, or whether things were in the light or the dark. And a lot of this is magic which your lab techs know. But if it's not written down, when somebody else reads your paper, they won't be able to reproduce your work. This has historically been an issue in synthetic biology, where some of the high-profile publications are presenting good work, which does work, but where the publication itself doesn't give you enough information to be able to do it again yourself. And remember, the thing about synthetic biology is we're trying to raise the bar towards an engineering discipline. Other people should be able to make it and reproduce your results, because we're trying to get to the point where this is technology, not research. So we have a multitude of existing file formats for communicating things about biology, and preeminent among those is probably GenBank. They range from things like FASTA, which is just a way to write down a nucleotide sequence, up to things like SBML and CellML, which are there to describe dynamic behaviors. There's a multitude of these formats which cover everything from Sunday. Most of these were created for the purposes of natural history. By that, what I mean is cataloging what is found within nature. And so there, your focus is upon the data collection, recording where things came from, where types could be accessed from in future, and that sort of base level recording of what you found. Synthetic biology isn't really about that. It's about forward design. So it's about planning to build things which don't exist yet. This means that there's a lot of information you would like to capture about your design, about its intent, about abstractions which you are using within the design to make it compositional or reusable. And these don't really exist within nature. So nature doesn't really produce biological components that have test harness hooks. Because nature tests things by killing off stuff. Whereas in synthetic biology, we do design things with hooks there to add in test harnesses. Because it's engineering, we want to test. These kind of things, to document them clearly, we sort of run out of expressivity in the existing formats, like GenBank. So back at about 2010, Herbert Sorrow identified the need for a better standard for documenting supporting materials for synbio design. His PhD student Michael worked with him to develop version 1 of SBOL, the synthetic biology open language, sometimes called the synthetic biology ontology language, for historical reasons. Since then, the data standard has moved away from being a one lab project to being an international, multinational consortium of contributors. It has an editorial board. We have regular international meetings to talk through design issues. We're up to version 2.2 now, and I think sometime next year or the year after, we're looking to do a version 3. So I'm going to very briefly talk about the major things within the ShortBOL data model. There are other talks you can find online which will go through UML diagrams and documentation in exquisite detail, but I'm not going to bore you with that here. This is just the 2,000 feet intro. So ShortBOL makes a distinction between the artifacts in your design, the physical stuff, which we call components, and composing them together in ways that have behavior, which we call modules. So an example of an artifact may be some engineered DNA, but it would also be the cell that DNA is within and the Petri dish that that cell is within. It's any physical stuff. We would also include things like sunlight or pressure or temperature. They're all things about the physical part of your design. And then modules are the aggregates of those which have behavior. So a promoter driving open reading frame, producing an enzyme which then digests a chemical, that composite with its behavior of those things being produced and catalyzing those reactions is a module. So the data models for components and modules have first-class constructs for composition and reuse. This is really getting back to what it is to mean to be doing designing rather than describing. So there's a hierarchy of DNA composition, for example. You can build bigger sequences out of smaller ones. You can build bigger modules by wiring together smaller modules. And you can stack these things up as far as you would like. And the composite behavior is, in some sense, the behavior you would get by gluing together all the behaviors of the parts. It supports composition in that you can take your component or your module and you can reliably use it in many other designs as a part within those designs. And the data model is designed so that the underlying knowledge about those small parts you're using doesn't change under you. So things are never edited with the same identifier. This really comes into versioning and revisions and identification. So all the entities within an ShortBOL design have a unique ID, they are versioned, and they're attached to a longer term persistent ID. It's not uncommon for you in the real world to make a DNA design, get it fabricated or clone it out of an organism you have in the lab, put it into your design and discover 6 months later that the DNA that was actually there wasn't the one you expected. And you then have to go back and find all of the downstream designs which used that DNA and then decide whether to throw them away or make them again with the new improved fixed sequence. Because everything in here is tracked and versioned and identified, it's a one-click search to find all the uses of your old broken component and then automatically come up with candidate fixed versions. Together with the data model, there's a standardized visual representation. So we have a standardized way to represent common parts of designs like promoters and open reading frames. This has had some adoption even outside the ShortBOL community. It's loosely based upon standard genetics diagrams, but again with some tweaks to meet the needs of design rather than description. The data model itself is captured within some UML documents and diagrams and a lot of supporting human readable text. I'm not going to show you these today as I said, but there are tools to turn those automatically into data models in your language of choice, or there are a number of libraries such as libShortBOL where people have by hand made libraries in your favorite language to manipulate ShortBOL data. There's also a representation of the data model as RDF. So there's an RDFS terminology, and you can describe everything in ShortBOL as RDF triples. This provides us with extensibility hooks. So if you have any data which doesn't fit the current ShortBOL data model, you can get it into ShortBOL, into your ShortBOL documents and the document exchange by virtue of encoding it as RDF and just linking to the objects we have. The exchange format is an RDF XML dialect, which can be read and written by any RDF compliant tools. And there are a number of online repositories now which allow you to import and export ShortBOL documents based both around RDF triples and based around more bog standard document blobs. So ShortBOL in the real world. ShortBOL has some use now within a range of tools, CAD tools. Some of these use ShortBOL as their native data model. Others support ShortBOL as an import and export format. There's active development of tooling and of more tooling to support ShortBOL. And there's 2 online repositories, which I should mention. The SynBio Hub at Newcastle University, which is an online ShortBOL document repository and triple store, and the JBait ICE installation for ACS SynBio, which is a one-stop shop for uploading documents for publication. So at this point, I'd like to run a PowerPoint presentation. This was recorded by Professor Chris Myers from Utah. He's one of the main ShortBOL collaborators.
This demonstration is going to illustrate a complete synthetic biology workflow using ShortBOL. In this workflow, we're going to design a genetic toggle switch using parts from the cello dataset that was created by Chris Voigt's lab at MIT. We begin by using the iBiosim software to create 2 genetic inverters to construct the toggle switch. So for the first inverter, we're going to add a promoter, and we're going to find from the cello parts a promoter that's repressed by the BETL protein. And then we need to add the gene that's going to be coded for. And in this case, let's select this one that codes for the AMTR protein. If you open that up, you can see that it includes a ribosome binding site, the coding sequence for AMTR, and a terminator. So that completes the first inverter. Now we need to move on to create the second inverter. Now the second inverter needs to have a promoter, which is repressed by the AMTR protein. And then we need to have a gene, which creates BETL. That completes the second inverter. Okay, so the next thing we'd like to do is build a model of this. So we select the 2 inverters to use in our design, select Generate Model, and select the symbiohubprogrambiology.org instance where the data is stored. Press OK. And now it's finding all the interaction information, namely what proteins repress which promoters and which proteins are coded for. Select OK. It's returned to us an SBML model that describes the behavior of our genetic circuit. So we can see that here's a model for inverter 2, which is an inverter that's repressed by AMTR, encodes for BETL. Here's a model for the second inverter, which is repressed by BETL and codes for AMTR. And then this one is a model of the composite circuit for the entire design that constructs the toggle switch. We're now going to insert this design into a larger design for testing and simulation. So we add this inverter into our testing structure, like so. Save that. And now we're going to simulate this. We're going to simulate it using stochastic simulation, we're going to perform 20 iterations of stochastic simulation and compute the average result that we get. And this is all done. We have a simulation like so, and you can see that it's acting like a toggle switch, namely it turns on when IPTG is applied, stays on when it's removed, and is reset when ATC is added to it. So this is a nice plot, so I think I'm going to save it. And save that plot for later. Out that goes. Now the next step is I'm going to save this design that I've just constructed into the SimBioHub data repository. So select Export SimBioHub and select the data repository that I want to export this to. Enter my password, provide some information about my design. Like so, and select Upload. And it's now going to upload all the design information I've just constructed, like so. And we go over now to simbiohub.org, and when I log in, and select Browse Design Collections, you can see that on simbiohub.org we have a collection for the AC Synthetic Biology Journal, a Bicellus collection, the entire IGM parts registry, the distribution for 2017, a couple ICE instances, and a link to a private repository within Benchling. We now select Submissions. We can see the genetic toggle switch that we have uploaded. It's in a private submission that only we can see. And here's all the various information we have about it. At this point, maybe I'd like to take that nice plot and add it to my design. So I can attach this plot. I can then describe this design. And I can add my plot to the design, like so, and save that into my little wiki, and you can see that I have that it's been added. So at this point, perhaps I'd like to do some sequence level editing. So let's select the first inverter. That brings up a page describing the inverter. You can see here's a diagram of that. Remember, it's composed of a promoter and this gene. Here's the substructure for that, and if you click on that, it's going to take me over to the Boston University instance of SimBioHub for the Living Computing Project, where the Cello dataset is stored, and you get a page that describes it, including the reference for the part, as well as some data that's attached to this part. Now we come back here, and we'd like to send this design to the Benchling software for further editing. So it's now copying this design into Benchling. And if we click on this, we can see what it looks like. Inside Benchling, you can see it's been flattened because Benchling only works on flat designs. And if I click Edit, it's going to open up a Benchling window, which allows me to do sequence level editing within the Benchling software. I can click on one of these annotations, for example, and I could change its name if I wanted, like so. If you come over here now and refresh the page, you'll see that the changes within Benchling immediately are reflected within SimBioHub. Okay, now I'm done with my sequence level editing, so I'm going to copy this from Benchling back to SimBioHub, and I'm going to copy it into my collection, like so. And so now this is showed up within my collection here, and there's the sequence level editing design. Now at this point, perhaps I've actually constructed this in the lab, and I want to keep track of the plasmid I've constructed, and I can use the ICE registry for that. Select Send to ICE, select an ICE instance, send it there. Now it's copying it into the ICE registry. And so here's the genetic inverter within ICE, on the ACS Synthetic Biology ICE. So here it is there, and if you come select there, you can open it up within the ICE instance, and you can view it in ICE. And if I'm happy with that, I can copy it from ICE back into SimBioHub into my existing collection like so. And I'm back to my original collection. Okay, so at this point I think it's pretty good and I'm ready to publish it. So I want to submit my paper and I want to share this privately to the reviewer. So I can click share, and this creates a shareable link. So if this link here is not accessible by anybody but me, but if I use the shareable link, just like in a Google Doc, and select that link, this can be opened by anybody. So if we pass that link to the publisher, then the reviewers can open up your design and see the design information about your design. Once your paper's been published, perhaps you'd like to add the reference into here, a PubMed ID, like so, and that will look up the reference for your paper. And if you select Make Public, you can then decide where you want to publish it. I want to publish it into the Synthetic Biology collection. Select Make Public, and it copies it over into that collection. And now I can open this here, and I can see the genetic toggle collection for my design, and all the design information has been published, and anybody can see it at this point and search upon it and use it in further designs of their own. And that concludes our demonstration.
Thank you, Chris, for that. So what is the future of ShortBOL? Obviously, there's a lot left to do. We would really like to expand the coverage of the data model. At the moment, we really capture the design intent. What's it built from? What do we want it to do? It needs to be extended towards measurement and fabrication, so to describe what people actually built, what the thing they actually built really did. And then to close the cycle around to capture what's going to be done to try and close the gap between the intent and the observed behaviour. We also need to push for extended community adoption, and I think this will really come from having compelling tools and compelling abilities. And so widening the range of tools that support ShortBOL is obviously a priority, developing novel tools which support ShortBOL, and engaging with tools developers to make sure that they have ShortBOL import and export capacity. If you've been inspired by this to get involved with ShortBOL, there's a number of things you can do. You can go to the shortbolstandard.org website, and from there there's information about how to sign up to our mailing list and other ways to get engaged with the developer community. There's also information on there about the standard, tools which use the standard, and so on. I would encourage you, if you've got a synthetic biology paper in the pipeline, to consider publishing in ACS SynBio. They allow you to upload your designs as ShortBOL, and they provide some tooling to help with that through SynBio Hub. So SynBio Hub is our online repository, which you saw as part of the demo there, developed by our PhD student James and others. You can sign up for a free account there and get going with browsing designs and uploading your own designs. Before I end, I'd like to acknowledge a couple of people. Chris Myers, again, thank you very much for the video. We have lots of collaborators within the ShortBOL community, both academics and commercials. A representative selection of those are in the image on the right. They include universities, research consortia, and some multinationals. So thank you very much for all the work that they have put in over the years in supporting us, both with money and with time and personnel. Thank you for listening to my talk today on ShortBOL, and I look forward to seeing you within the ShortBOL community